About the next topic, that is the transduction. Transduction, it is the gene transfer takes place into the bacteria from donor to the recipient cell via bacteriophage. This is a vehicle. There is a requirement of the vehicle. Means bacteriophage acts as the vehicle which takes the DNA or segment of the DNA from donor cell and it transfers to the recipient cell. So that this kind of the technique is known as the transduction. In previously we discussed transformation when next DNA molecule, next foreign DNA molecule, it is get transferred into the competence cell, that is the transformation. Then into the conjugation, the requires the physical contact from donor cell to the recipient cell. Transfer of the gene or DNA takes place by physical contact. Here, this is the another technique, transduction. Transduction, it is a transfer of DNA from one bacterium to another via bacteriophage. Okay, donor to the recipient cell. And this bacteriophage is the virus specifically attached to the bacterial cells. You all of you know it is a virus of the bacteria, bacteriophage. It is composed of the genetic material surrounded by the protein coat. It undergoes two types of the life cycle: lytic life cycle and lysogenic life cycle. All of you know this. Those phages which or those bacteriophages which multiplies via lytic life cycle are called as the virulent phages. Okay, and those phages which multiplies via lysogenic life cycle, these are called as the temperate phase. So you always remember about the virulent phases and the temperate phases. So we are going to see here the steps in the lytic and lysogenic life cycle. In lytic cycle, how which are these steps? The first step is the adsorption. Here, the adsorption of this bacteriophage takes place onto the host cell. Generally, you can see this is a phage, this is a DNA, red color DNA, and phages are made up of only two components, as the viruses are made up of two components: the protein coat and the genetic material, that is the DNA or RNA. These are the bacterial viruses, these are the DNA viruses. They contains DNA as the genetic material and the protein coat. These are the phages. They consist of the parts like the head maybe the icosahedral head. This is the sheath, tail fibers discussed in Hershey and Chase experiment. Then first step is the adsorption onto the bacterial cell surface or cell, generally outer membrane is the cell wall, and onto the cell wall the receptors are present onto the this cell surface of the bacteria, and on these receptors these cell fibers and sheath attaches, and this attachment, it is initially reversible, and then after some time it becomes irreversible. If the phage of that particular strain, particular species, then it becomes irreversible. It is permanently gets attached onto the surface. So this step, it is known as the adsorption. Second step, virus injects the genetic material into the cytoplasm of the host. Integration of this genetic material, it is takes place into the host cell, and after that, okay, after that, it enters genetic material of the phage enters into the uh, cytoplasm of the host cell, and because of that into the cytoplasm, intracellular development of these phases takes place. They uses the host machinery, that is they uses the replication machinery. They uses they synthesizes their proteins, the viral proteins or parts proteins. There they includes the coat protein, then sheath protein and tail fibers. And in this way, they synthesize all the proteins which requires for the making of this virus or making of this phage. And after that, assembly of this phage takes place. And the assembly of phage means that here the coat protein, in that coat protein, the phage genome is um, packed, and after that sheath attaches to the coat protein, and then after that the tail fibers are attached to this uh, sheath, and then in the tail fibers contains the enzymes which disintegrates the cell wall and cell membrane. Okay, this after the disintegration of the cell wall and cell membrane, the release of this phages takes. And after from the one infected host, they are get released into the environment. Means आता येतोय का? These viruses then after release, these viruses it again infects the new healthy cells. In this way, one lytic cycle, the millions of the phages are produced. Here shown very few, but the millions of the phages are produced in the one lytic cycle or in one host cell. Then,
हो आता येतोय.
then the next lysogenic cycle, how this phase multiplies. First step is the adsorption. Then second step is the injection. And after injecting his DNA, phage DNA, it integrates into the host genome. This implies via lytic life cycle, but temperate phases. After the injection of his genetic material, integration of the viral genetic material takes place into the this host genome, and this step it is called as the lysogen life cycle. This phases multiplies via two uh, kinds of the life cycle: lytic life cycle and lysogenic life cycle. So the phages here in the transduction study, which phages are taken? Phages that transfer the donor to the host. P22, which infects Salmonella typhimurium, and both are the here phage visits to or get adsorbed onto the host cell. Okay, then phage infects the bacterial cell and injects its genetic material into the host. And in lytic life cycle, for probability of 10 raised to minus 5 to 10 raised to minus 6, 1 into 10 raised to minus 5 or 10 raised to minus 6, 1 into 10 raised to 5 phases or 10 raised to 6 phases, this probability. So this bacterial host genome may get packed into the, hence this phase, it is known as the transducing, inserts this host genome of the one cell. Again, they will infect the another healthy host. So integration of this first host genome takes place into the another host. Here, in this segment of the DNA is from the first host, and it integrates this into the another host. Suppose this host is a lysine or histidine minus, and this host it is get converted into histidine plus. Like this kind of the transduction takes place generally, and this type of the transduction we can call is the generalized transduction. This means the transduction which takes place by the virulent phages. We can say it is called as the generalized transduction. Any piece of the bacterial DNA can be incorporated into the phage head. Any piece of the bacterial DNA can be packed into the phage hose, phage head, and then random. It is a random. Hence, it is called as the generalized transduction, not the specific. Any kind, any fragment may get packed into the this phase. Hence, it is called as the or this phase transducing particle. It is called as a transducing particle. And then when it infects the new host, new healthy host, it transfer the that segment of the DNA which was packed into his head, that it transferred into the another bacterium. And in this way, the gene transfer takes place from the one host to the another host, from the one donor to the another recipient cell. This way, this process it is called as a generalized transduction. And transduction was discovered in 1952 by the scientists Joshua Liederberg and Norton Ginder. They used